All right, let's talk about the next step in talking about digestion. What actually happens at each step of the process? So there are 4 main steps with food consumption for heterotrophs like animals: ingestion, digestion, absorption, and elimination. And we'll talk mostly about the first 3 of those. Ingestion is the taking in of the food, whatever it is, whether we're talking about filter feeders or bulk feeders like we are. And there's quite a few different mechanisms for this. So filter feeders, which we talked about a little bit, like for example, for species that have a lophophore feeding structure, that's one kind of filter feeding. Things that, and even, we have a baleen whale over here, things that sift through particles in water. So filter feeders are all aquatic species. Substrate feeders, these are organisms that live on their food. So many insects would qualify as substrate feeders, and parasites would also be substrate feeders because parasites live on or in their food. For example, fleas that live on on a mammal, those would be substrate feeders, and also ectoparasites. Fluid feeders drink fluid, and this could be from a plant like these aphids here, or from animals like ticks here. And if you're interested, these 2 ticks are the same species. They're both female ticks. This is post blood meal, how expandable they are. It's kind of, we are bulk feeders, so our almost all mammals are bulk feeders, with the exception of maybe vampire bats are the one example I can think of that would not be a bulk feeder. So bulk feeders consume fairly large pieces of food, which is exactly what we do, and then digest it internally. Digestion is the process of breaking down the food. And there's 2 types of digestion. One is called mechanical digestion, and this would start with things like chewing, but also the mechanical digestion that occurs in our stomachs, and also in other animal stomachs, like in bird gizzards, physically grinding up the food into smaller pieces, increasing its surface area, making that surface available for enzymatic digestion. And then chemical digestion is using enzymes to break down the actual molecules, large molecules being broken down into small molecules, like proteins being broken down into amino acids. That would be chemical digestion. The next step would be absorption of those nutrients into the body. And then, and we'll talk a bit about that in the small intestine. And then elimination would be getting rid of whatever cannot be digested. Animals have specialized compartments. Even the very simple animals like nematodes that have a very rudimentary alimentary canal, they still have specific regions of that digestive tract where chemical digestion and absorption take place. And so we'll talk about the different parts of the mammalian digestive system more specifically. Some of the more primitive animals like sponges and cnidarians and even flatworms have what we call intracellular digestion. They absorb particles of food not by breaking them down and then absorbing the nutrients like we do, but by engulfing those small food particles in vesicles and then bringing those vesicles inside of the cells and then digesting them that way using lysosomes fusing with them and peroxisomes fusing with them. This is a very primitive kind of digestion. This is what many single-celled protists use as their method of eating, like amoeba do this. Sponges entirely do this. But we also have some cells that are still phagocytotic and eat small particles by bringing them into vesicles, like some human white blood cells, part of our immune system, like macrophages and natural killer cells, which actually consume particles, bringing them into vesicles that are then later broken down. So here is Cnidaria and flatworms doing this. So sponges do this as well. So they do secrete digestive enzymes into their gut. Their remember that Cnidaria and platyhelminths have a single opening gut. They have a mouth but no anus, so no alimentary canal. They just have a sac-like gut. And they do some chemical digestion in those guts. So it's kind of a mixture of primitive sponge-like absorption of particles by phagocytosis and digesting outside the cell like most of the rest of the animals do. They kind of do a little bit of each. So large food particles are engulfed by vesicles, endocytosis, and brought into the cells for further digestion, but they also do enzymatic digestion. What we do is called extracellular digestion. So all of the digestion of our food takes place outside of cells, and only the individual nutrients are absorbed and brought into the cells. And really, the interior of our digestive system, the lumen of our digestive system, is outside of our body. We have a continuous epithelial layer on the outside and down inside of our digestive tract all the way through. So food that you eat stays outside of your body. It's outside, it is digested, and then only the nutrients are absorbed. Things that we cannot use, like plant sterols, for example, remain in the lumen of the gut and are not absorbed into our actual body in any way. So once we get past the flatworms and the acials, and pretty much all the rest of animals have a one-way gut, an alimentary canal that has a mouth and an anus. Food only comes in the mouth, waste comes out the anus, and there are specialized regions along the way for digestion and absorption, and specialized cells lining the gut that either secrete enzymes or can absorb nutrients. And we have accessory glands that secrete these digestive enzymes. Some enzymes are secreted by the small intestine itself, but there are other digestive enzymes and liquids and acidifying agents that are secreted by other accessory glands. So the salivary glands, that's where digestion starts, is in the mouth with chewing, and saliva also contains amylase, so the breakdown of starch begins in the mouth as well. The pancreas, one of the functions of the pancreas is kind of a two-function organ. The pancreas secretes hormones into the bloodstream, like insulin, and it also produces digestive enzymes that are secreted into the small intestine. The liver makes enzymes and also bile that helps digest fat, which is stored in the gallbladder. If you don't have a gallbladder, that's okay. You just can't secrete a huge amount of bile at one time. Bile is still made and is just continuously secreted into the small intestine in the case of people who are missing their gallbladder. The gallbladder is just a storage organ. It doesn't make any enzymes or emulsifying salts itself. Those are made by the liver. So food processing starts in the oral cavity, which is the mouth. Salivary glands wet the food and assist with chewing. By lubricating the food, that makes it easier to swallow. If you don't have enough saliva, food is difficult to swallow. Mucus is a mixture of proteins and water, and this also helps the glycoproteins, the proteins that are in mucus, are very hydrophilic and bulk up with water when they're exposed to water after secretion. And saliva contains an enzyme amylase, which starts the breakdown of starch. So if you chew starchy things long enough, and I mean like a really long time, like several minutes, they actually start to taste sweet because you're releasing the glucose that's in that starch. So we'll talk a little bit more in detail about all of the parts here of the digestive tract. So food, after entering the mouth, goes into the pharynx. And oh, here we see this word again, pharynx. So the pharynx is before the trachea splits off. So the pharynx can contain both air and ingested food. The esophagus, however, is only going to contain swallowed food. The esophagus doesn't do any digesting or processing of the food. It's just moving it down to the stomach. The stomach, we'll talk about what the stomach does. We have then food goes into the small intestine. The small intestine is where the vast majority of both digestion and absorption takes place. And then the food moves into the large intestine, which is mostly about absorbing the water, and a few vitamins and things that we'll talk about too, and then elimination. So we have a problem that not all mammals have, and that is that there's a chance that food is going to end up going down the trachea. So the pharynx is up here. The pharynx is where you can have both air and food, and then these two compartments are separated. And there's a little flap of tissue, the epiglottis, that prevents food from going down into the trachea. So it closes when you swallow. The swallowing reflex closes the epiglottis, and food then can go into the esophagus and not into the trachea. There's also a little sphincter here at the top of the esophagus that prevents air from going down the esophagus. So when you breathe, you're not going to be sending air into your stomach. Now, some people can learn to control this a little bit, and these are the people who can burp on command. They have trained themselves to be able to suck air down into their stomach deliberately. So that's something you can work on for when parties start happening again. So once the food reaches the stomach now, it has saliva mixed in with it. It's been mechanically chewed a little bit. And there's some amylase in there that's already starting to break down the starches that might be present in the food that you're eating. The stomach secretes a mixture called gastric juice, and once this gastric juice is mixed with the food, the contents of the stomach are called chyme. And there are two main types of cells here that are important for digestion, the chief cells and the parietal cells. And they are both found in these gastric glands, which are all over the inside of the stomach. There's also mucus-producing cells, and the mucus that's produced in the stomach is what protects the lining of the stomach from being digested itself. So the chief cells produce an enzyme called, well, this is the precursor. Pepsinogen is the inactive form of the enzyme. 44 amino acids get cleaved off of it, and then it becomes the active enzyme, pepsin, which breaks down proteins. Parietal cells are what makes the stomach acidic. Parietal cells secrete hydrochloric acid, and it's the hydrochloric acid that helps activate the pepsinogen. And so without an acidic environment, the pepsinogen wouldn't be able to even be converted into the active form pepsin. And even if it was in the active form pepsin, it could not function. Remember that enzymes have a pH at which they are optimally functional, and the optimally functional pH for pepsin, for human pepsin, is about a pH of 2. So if your stomach is not acidic enough, pepsin cannot function, and it won't even be cleaved into the active form most of the time. So The parietal cells are secreting hydrochloric acid. Chief cells are secreting pepsinogen. The pepsinogen gets activated by the acid. Mucus cells are secreting mucus that is lining the stomach and protecting it from its own digestive juices. So pepsin is what we call a protease, meaning that it breaks down protein. And the hydrochloric acid has several functions. One function of the hydrochloric acid is to kill bacteria, and this is something that is seen commonly in carnivores as well as some omnivores, like bears as well have an acidic stomach. Kills bacteria that might be ingested with the food, but of course, as we know, not all of them, because you can still consume bacteria in your food that will make you sick, like salmonella. So these bacteria are not killed by the stomach acid, but most of the bacteria that you ingest with your food are killed by your stomach acid. It also denatures proteins, so pH is one of the things that denatures proteins. So just the acidic nature of the gastric juice is already helping to unfold these proteins so that the enzymes can get at them and help digest them. And this is a really nice microscope image here of one of these gastric glands. So you could kind of see this very thin opening here. You can barely see it, this thin opening. And CC is for chief cells and PC is for parietal cells. And you can see that they are clearly visible as different kinds of cells lining this gland. I don't see any mucus cells in this particular slice. This must be a slice further down where there aren't any or not as many mucus cells. So the small intestine is the longest compartment, and that's because this is where almost all of the absorption is taking place. So the stomach is not absorbing any nutrients. The esophagus and the mouth are not absorbing any nutrients, except things that can just dissolve across the membrane, like alcohol is absorbed in the mouth and in the stomach, but that's not really a nutrient. All of the nutrients are being absorbed in the small intestine, and then a few are being absorbed in the large intestine, which we'll talk about that. So, and further hydrolysis, all 4 categories of macromolecules are digested in the small intestine as well and broken down into their constituent molecules. So having a huge surface area increases the efficiency of the small intestine. So we have an enormous surface area. If you took the surface, the membrane surface of your small intestine and spread it out, it would be about the size of, I forget what the, half a football field, something like that. And it's thanks to 2 things, villi and microvilli. So I'll show you an image of that. So this scanning electron microscope image here is of microvilli. So this is one cell. This is the surface of one cell. You can see the edge of the cell here. And then these membrane-covered microvilli are all covered with membrane. So that membrane has greatly increased the surface area for absorption. This microvillar surface is called the brush border in the small intestine because when scientists first saw it under the microscope, it looked like a hairbrush. So that's why it's called the brush border. The villi are made up of many, many cells, and these are big enough to be seen with the naked eye. So if you see the interior of the small intestine, and if you're going to go on to anatomy and do cadaver dissection, you will do this. If you cut open the small intestine and look at it, you can see the villi with your naked eye, and they look like tiny, tiny little fingers in the small intestine. And if you look microscopically at the individual villi, so we have this little square here blown up, each individual cell in the small intestine is covered with microvilli. And so the surface area of our small intestine is enormous. And nutrients are not brought in vacuoles by phagocytosis. Instead, it's entirely absorbed across the cell membranes using transport proteins. Some of them just diffuse across the membrane, but most of the nutrients are brought in by active transport of some kind. So all the carbohydrates and amino acids are brought across the membrane entirely by active transport. And for some of the fatty acids, it's not active transport, but most of the nutrients, it's active transport. And then it goes into the bloodstream and then to the liver by the hepatic portal vein. There's a little video here about a little review video about membrane active transport, which was covered in Bio 1151. But if you need a little refresher, I'm not going to show it to you right now, but it's in the PowerPoint and you can watch it. It's like 3 minutes long. So inside these intestinal tracts of animals, many animals, including us, have a mutualistic relationship with our gut bacteria. Ours is different from many other animals. Herbivores seem to have the extreme version of these adaptations. So herbivores have fermentation chambers in their gut, and sometimes it's in the beginning of the gut, in a compartment of the stomach, and sometimes it's towards the hindgut in a compartment called the cecum, and we'll talk about both of these. Some animals pass food around more than once to make sure that these symbiotic bacteria do their digestion, and then the food goes back up to the small intestine for nutrient absorption. And the most elaborate adaptations are found in the ruminants, and I'll show you both of these kinds of gut adaptations for digesting the food. So here's a rabbit which has hindgut fermentation, and this adaptation allows these organisms to survive on a diet that is very, very high in plant material, almost exclusively plant material, and that has a huge amount of cellulose in the diet. So the animals themselves cannot digest cellulose. It is their symbiotic bacteria that are living in their gut that can digest the cellulose. So here's a rabbit's digestive system, and right at the beginning of the large intestine is the cecum. This is where all of those bacteria live. They are anaerobic bacteria that can break down cellulose. They make more bacteria using that cellulose, and then the bacteria are consumed by the rabbit. So these bacteria can go back into the small intestine. The bacteria are broken down by digestive enzymes, and the fatty acids and the proteins in the bacteria are absorbed. And then it goes back to the large intestine. So they actually get a lot more from eating grass, let's say, than a human would, because we don't have this kind of fermentation in our gut. Although a little bit of the cellulose in our gut does get broken down by bacteria. Ruminants, which include cattle, sheep, goats, bison, antelope. There's a huge list of these animals. So ruminants have a separate stomach compartment, the rumen, for these fermenting bacteria. So when a cow eats grass, that grass goes into the rumen, where it remains for many, many hours. And the bacteria in the rumen break down the cellulose and make lots and lots of bacteria. Then that food goes back up to the mouth, and they chew it some more, and then it goes and is finally digested. And that all of the bacteria that go through this process get digested and consumed. So cows actually have a high fat diet because most of their calories that they are absorbing in their small intestine are coming from the fatty acids in the bacteria. So they're not really eating the grass. The grass is feeding the bacteria, and then the cows are eating the bacteria. So and rumens can take the worst forage, the least nutritious forage, and make use of it because they have the best and most efficient use of these fermenting gut bacteria. So land that we can't grow food for humans on, that's too dry for growing crops, too poor, the only thing that grows is grass, cows can turn it into a huge 1,000-pound body because of the rumen. It's really an amazing evolutionary adaptation. So once food is mostly absorbed and digested, it goes to the large intestine in mammals. And the large intestine includes the colon, the cecum, which our cecum is really tiny compared to other animals, and then the rectum, and then the rectum leads to the outside, to the anus. So everything that can't be digested or absorbed ends up eventually being eliminated. The cecum in many mammals is where fermentation of plant material occurs. So mammals that are either completely herbivores or are mostly herbivores that aren't ruminants, they have an enlarged cecum. So horses and rabbits fall into this category, and as well as most of the other primates. So chimpanzees, gorillas, monkeys have an enlarged cecum. Even some marsupials like koala bears have an enlarged cecum, and this is where fermentation of cellulose is occurring in those animals. And they vary a bit in how much of the cellulose actually gets broken down. So for example, chimpanzees are less efficient than some of the monkeys, and they still have way more fermentation that occurs than what occurs in a human gut, but they're kind of halfway in between some of the very efficient herbivores and what the human gut or a carnivore does. Our cecum is mostly completely shriveled up, and that's what we call the appendix, and it's considered vestigial. It's a vestigial cecum. So a vestigial structure is a structure that was used in an ancestor, but isn't used in the organism in which we currently find it. So we have several vestigial structures. One of them is our cecum, which is the appendix, which people have tried really, really hard to find a use for the appendix because, well, mostly that's creationists, because it goes against their hypothesis for a human to have a useless structure, and especially a structure that was obviously useful in ancestors and is still being used in almost all the other primates. But they have all failed. The appendix has no function, and in fact, as many millions of humans know, you can have your appendix removed and it has no consequences whatsoever. So here's the human small intestine and the beginning of the colon. There's a little blind pocket here, which is what we call the cecum still in humans, although it doesn't house any anaerobic bacteria that can break down cellulose. And then we have this little hollow finger called the appendix. And when you get appendicitis, what happens is an infection develops inside here because the appendix is a hollow tube that's open to the intestine. So it can get inflamed and infected. And in some cases, it's been shown recently that just antibiotics can help. And often it needs to be removed to avoid a reoccurrence. But here's our large intestine. It has 3 portions here, ascending, transverse, and descending. And this, there is a little bit of variation. 99% of people have a large intestine that looks just like this, but there are, occasionally people who have extra loops in their large intestine, or have a really exceptionally large or long large intestine. Most of the time, they would only find out accidentally. So what does the large intestine do? Mostly it absorbs water, although it does house most of the bacteria. So most of our intestinal bacteria are here in the large intestine. And they do break down about 10% of the cellulose that you consume. So if you consume a huge amount of plant material that's full of cellulose, about 10% of it is going to get broken down by these colonic bacteria. This just means bacteria in the colon. And those bacteria are going to use that glucose for their own energy. We don't get any of the glucose. The large intestine does not absorb any glucose. But they do make some useful vitamins. So they make vitamin K and 2 B vitamins, thiamine and riboflavin, but not vitamin B12, you'll notice. We don't have the right bacteria for making vitamin B12. All of the fermenting organisms, all of the ruminants, and most of the cecum fermenting mammals have bacteria in their cecum or in their rumen that make vitamin B12. We don't. We make 0 B12 in our intestines. And a few other metabolites, things like butyric acid, which the bacteria make from broken down fatty acids that aren't previously digested. And the butyric acid is actually used by cells in the large intestine as a fuel source, as it's a metabolite that actually feeds your large intestine cells. So these vitamins are absorbed in the large intestine and also about 90% of the water. So the contents of the small intestine are extremely liquidy and watery. Once they get into the large intestine, as they move along and more water is absorbed, they become more and more solid. So this is a water conservation measure. It allows us to have to drink less water because it's all being absorbed. There's some salt balancing that happens too. Excess potassium can be excreted into the cavity of the large intestine. So there's a few things like that too that help us balance the, well, I'm calling them salts, but you may have heard of them called electrolytes. Our large intestine does a little bit of that as well. So this is a summary of the digestion of each of the 4 macros that are the 4 major macromolecules that takes place. So in the oral cavity, pharynx and esophagus, really there isn't any digestion taking place, but it starts in the mouth. Carbohydrate digestion is the only thing that's occurring there. Starch is getting broken down thanks to salivary amylase into short polysaccharides. One of those short disaccharides is maltose, which is 2 glucose stuck together. The disaccharides, nothing is happening with the disaccharides yet, like sucrose and lactose in the mouth. Now we go to the stomach, and now we have further digestion. Disaccharides are going to be broken down. The stomach is where, if you as an adult can still digest lactose, your stomach is where that digestion is going to occur. Smaller polysaccharides that are made out of, like starches, are going to be broken down here as well. Pepsin is going to start breaking down proteins, not completely to amino acids. Pepsin only cleaves proteins between certain amino acids, but it's going to break down proteins, which some proteins can contain thousands of amino acids. Pepsin is going to break those down into little chunks. So now they're moving on into the small intestine. Instead of as huge proteins, they're moving on as little peptides that can be further digested in the small intestine. Okay, small intestine, there's 2 slides here for the small intestine. All 4 of our large macromolecules that we learned about last semester are digested in the small intestine. So enzymes from the pancreas that contribute to that. There are enzymes from the pancreas that break down protein, trypsin and chymotrypsin, break down these peptides completely into amino acids. Carbohydrate digestion keeps going from what was started in the stomach. There are also nucleases that can break down RNA and DNA, because of course all the food that you eat is from living organisms that had RNA and DNA, because that's what we're eating. We're eating other organisms, and that is not going to go to waste. The individual nucleotides can then be absorbed and reused. Although we can make nucleotides if we need to, our bodies can make them. We don't have to get nucleotides from our food, but they don't go to waste. Why resynthesize them if you can absorb them from your gut? Fat is digested here. The pancreas makes lipase, which can break down triglycerides into individual fatty acids. So glycerol is that 3-carbon sugar that's the backbone for triglycerides and phospholipids. And those can be then absorbed directly in the small intestine. This is what enzymes that are secreted directly by the intestinal epithelium can do. So they are secreting enzymes that can break down disaccharides like sucrose and lactose. They are secreting enzymes that are breaking down protein, and more of these nucleosidases that can break down nucleotides into their constituent components for absorption. Okay, so in the next video, the last video for this chapter, we'll talk about the hormone control of digestion.